Okay, thank you so much for inviting me. I don't know, you can hear me?
Oui, on entend.
Okay, great. Okay, thank you. First of all, thank you, the organization committee, that for this invitation and for this conference. Today, I would like to talk about multimodal neuroimaging of developing brain. So, one of the things that makes the brain such a complex system to try to understand is that it operates simultaneously on many different scales and many different levels. So there is spatial scales, and on the left image you can see, and here you have processes in the brain that are happening at the molecular level, going all the way up to the level of centimeters and tens of centimeters. So, so we have a kind of spatial scale. And there's even higher spatial dimension or higher spatial scales that we could talk about, and that's things like how your social environment or your cultural context can actually change your thought process and other processes that are taking place in the brain. So there is also temporal scales that there are things happening in the brain that are really, really fast at the level of milliseconds. So there are other dynamics in the brain that happen on the order of seconds to minutes and even hours. And of course, there are changes in the brain over the course of lifespan. Not to mention at the evolutionary scales of tens of thousand years. So we have also a kind of temporal scales, and there's many other ways of describing the complexity of the brain. So in the left image, you can see the right image, sorry, you can see the different brain imaging techniques that each of them can measure the brain activity in different scales. Here, I talk about EEG and functional near infrared spectroscopy. So the brain is a dynamic system, the factor shaping brain. There are three principal drives to form brain development. First is innate drive signal expressed via gene transcript. The second factor is environmental interaction, and the third one is the spontaneous neural activity. So we interact with the environment through our sensory and motor system, and this allows for these endogenous pattern of activity to be formed by the stimuli that we encounter. So the sensory motor experience is the way that we actually use our body. So this is particularly interesting to consider, but our knowledge is kind of limited. These three factors become especially interesting and important to consider when we think about how the brain responds to injury. For example, we might imagine that that when the brain is injured, then the dynamic system of that network is perturbed in some way, and this is very likely is going to alter the way these ongoing pattern of activity are shaped by interaction with the environment. So EEG in preterm shows a functional immaturity of the brain. The complexity inside the EEG in this period changes in the course of development. We can see different activity during sleep stage in different gestational ages. EEG amplitude is higher, and burst discontinuity is longer in younger preterm. And also the discontinuity suggests that some generators are modified with development. Some features are specific to neonates, such as the temporal, theta, delta brush, or frontal transient. So if we globally look at to the pattern of these activity from extremely prematurity to term neonate based on gestational age, EEG have specific maturation features. There is a dynamic in this maturation, which is characterized by, for example, EEG amplitude and discontinuity gradually decrease based on increasing age. There is continuity of different generators, which appear and disappear in this period of early infancy. This shows the kind of dynamic of the brain maturation. To see why there are these changes in EEG, we can look at these dynamics at maturation of the brain at three different levels: macroscopic, mesoscopic, and microscopic. And the macroscopic level, the brain has a gradual gyrification or its development. For example, this is important for the orientation of dipole. However, this may explain in part some aspect of EEG change of newborn, but the gyrification alone cannot sufficiently explain the dynamic changes of EEG. At the mesoscopic level, one of the possible cause of dynamic change in EEG is due to subplate. Here you can see subplate in yellow, and cortical plate in green, and some afferent fibers. Subplate is transient zone observed almost only in premature when thalamocortical afferent are waiting before to invade the cortical plate. From 28 to 32, 33 weeks gestational age, there is a massive relocation of afferent fibers from the subplate to the cortical plate, concomitantly with the elaboration of dendritic trees and the pyramidal neurons. Thus, the processing of external stimuli here in our case, I mean, it's auditory in preterm infant, is based on two circuits: a transient circuit involving subplate neurons and a permanent circuit within the cortical plate. Thus, our goal was to probe functional efficacy of these imager organization to process auditory stimuli. So if we look at macroscopic level of a neural and vascular network, both vascular and neural network are immature. Density of neurons and vessels is less in preterm than in adults, but the level of immaturity of each network seems kind of well adapted. So let's go to see the interaction between these two networks called neurovascular coupling. So why we use the EEG in NERS in preterm neonates? In fact, first is to see the electrical and hemodynamic response of the brain. And also, if we look at the brain imaging system in preterm neonates, it can be divided to two categories: structural and functional imaging. It consists of ultrasonography, MRI, and CT, are the imaging tools which used to look at the brain structure. Mostly cranial ultrasonography used. Among all the available functional imaging system for now is only EEG and FNERS that can be used due to kind of ethical restriction. There are some also MRI, fMRI studies that has come up. So EEG record excitatory postsynaptic potential and nurse measure blood oxygen saturation and concentration changes of the brain. So let's see what is the difference between these two kind of methods. EEG records electrical activity of pyramidal neurons, which is parallel and synchronized in resolution of millisecond, while NIRS record hemodynamic response of neuron and astrocyte, which doesn't really require necessarily parallel and synchronized. So this is one of the kind of main difference between these two techniques. Neural activity changes in the brain tissue are coupled to changes in cerebral blood flow, refers to as a hemodynamic response. And this is hemodynamic response is based on kind of basis of functional NIRS and MRI as well. So how NIRS measures these activities and how we can look at the brain activities. So in biomedical optics, we can have it, light and tissue can interact in two ways. Light can modify tissue, or in our case, the tissue optical characteristic can change or can modify light intensity. So here, for example, you can see the kind of brain, schematic of brain, skull, CSF. One near light source in red and one detector in blue. Near infrared light interact with the medium in two main processes, as a scattering or its kind of absorption. Some photons are scattered and some photons absorb. Some scattered and travel through the medium, and the rest of the photon detected by the detector. So the detected photon path is a kind of a crescent or a kind of banana shape. So because the scattering is relatively constant, when hemoglobin concentration increased, for example, more absorption, the detected light decreased. And when the hemoglobin concentration decreased, meaning less absorption, the detected light increased. And to calculate two unknown parameters, I mean, deoxy and oxyhemoglobin, two laser wavelengths required. The amount of oxyhemoglobin and deoxyhemoglobin can be achieved using a modified Beer-Lambert law. We come back to see how vascular network responds to neural activity or hemodynamic response. So two types of changes can be restudied using near infrared light. Kind of fast effect within the 50 to 500 millisecond latency, and slow effect with 2 or 10 second latency. These two effects have different origin that can be distinguished because of their larger difference in time scale. For example, fast effects are presumably related to the change in neural tissue, while slow effects have a kind of hemodynamic origin. So if we want just to look at these slow effect, neurons, like other cells, require energy and oxygen. This energy is supplied in the form of glucose and oxygen. Neurovascular coupling includes the following events. In fact, it's the neuron consume energy, oxygen and transport to neural tissue via oxyhemoglobin. There are decrease in oxyhemoglobin, and then, in fact, because of the nitric oxide or other neurotransmitters such as acetylcholine released by active neuron, cause a kind of relaxation of smooth muscle in arteriole, kind of vasodilation. This increase the kind of oxyhemoglobin and the kind of blood flow through the tissue. So we need the external stimuli or endogenous stimuli to see this hemodynamic response in our case. And so it can be ongoing spontaneous activity, auditory stimuli, or epileptic spikes that can induce this hemodynamic activity. So in one of the studies, we test the neonates. Test consists of 108 blocks, and each block contained 20 seconds of auditory stimuli by 40 seconds silence. And the syllables are ba and ga, and we have a kind of difference in voice and difference in phoneme between male and female. So in EEG, we look at the kind of trial, in fact. So special optical probe specifically designed to fit comfortable preterm head. It consists of 16 emitters and 2 detectors in each hemisphere. It covers a kind of pre-Stellman area. In black, you can see the position of the detector, and in red, the emitters. In between, there are 10 acquisition channels. So they are projected, in fact, is on the 30 weeks gestational age old preterm head. So here is the auditory hemodynamic response. There are increase in oxyhemoglobin in red and decrease in the oxyhemoglobin in blue for left and right hemisphere. We first demonstrate that the neurovascular coupling was sufficiently mature at this age to follow our stimulation paradigm. And the stimuli induced activity notably in posterior part of planum, then anterior channel. So this confirmed relative maturity of neurovascular coupling. So let's see this dynamic of this processing. The video is oxyhemoglobin concentration changes for different condition. Here you can see one sample nurse channel in the left and dynamic of HPO topography map in right for standard deviant and deviant voice and deviant phoneme. So we can see, in fact, kind of difference in hemodynamic response due to different conditions. Regarding to EEG recording, for recording high resolution EEG, different size of cap was made. We record 19 healthy preterm. And in fact, due to invisibility of 3D reconstruction of electrode position, we performed the kind of procedure to do it outside. In fact, but we first mold the preterm head using a silicone paste, then mold was filled with the plaster. Next, it was then covered with the cap, and at the end, electrode position kind of digitized. So in auditory event potential for each syllable, we can see one component. It shows as early as 28, 32 weeks gestational age, neural network are able to synchronize in such a way to produce an ERP, event-related potential. So each syllable induces an ERP with frontal positivity and posterior negativity. We can see also habituation to repetition of the syllables and so on. So here is the kind of mismatch negativity. We can see the mismatch response. Green line is the ERP response to the central EEG channel, and blue is the deviant voice. We can see two different mismatch in the response to change in voice has a different topography than the response to change of phoneme, revealing that the voice and phoneme can be coded in premature neonate by different neural networks. So you can see the first mismatch deflection occurs around 340 milliseconds, and kind of the second mismatch deflection recorded is about 600 milliseconds. In other studies, in fact, we want to see the kind of the value of the multimodality. So using EEG and NERS. So we know that the neonate born premature have a risk of intraventricular hemorrhage, IVH, and that makes a risk factor for cognitive impairment. Children with delay in language, I mean, these kind of children have a delay with language and speech. So if we can detect this inability as early as possible, then we are in the better position to treat more efficiently these disorder. So as early as possible, it's better, definitely. So using sonography, hemorrhage have been graded from 1 to 4 according to the degree of bleeding. We recorded and compared neonate with severe IVH, mean grade 3 and 4. If we look at the ERP response, these ERP have the same characteristic pattern for both groups, consisting of 4 positive deflections in frontal area. Figure shows that the difference of peak latency between healthy and the IVH subject. There is a kind of significant difference between latency and between a healthy and IVH subject. However, is the kind of that if we look at the amplitude and peak, the significance is not so much extreme. So about the hemodynamic response, this figure shows the comparison between healthy and IVH group in oxy and deoxyhemoglobin. Premature infant with IVH showed less pronounced hemodynamic response to auditory stimuli for both hemispheres than healthy preterm neonates. So the hemodynamic response is less in IVH than healthy neonate. For all condition or area under curve or within the first 10 seconds of stimulation, significantly smaller in both hemispheres. Here, in fact, I would like to emphasize the value of multimodal recording and its value for clinical application. So while EEG somehow cannot detect, it's kind of blind to some aspect of neural and neurovascular coupling, neural activity, using kind of complementary method, we can go further and we can have a better sensitivity to specific pathology. So yeah, here is just you can compare the dynamic of oxyhemoglobin changes in healthy and IVH, and the kind of the richness of the hemodynamic response in the healthy compared to the IVH when we record the hemodynamic response. Okay, thank you for your attention, and I'm available if you have any questions.